In this topic, we're going to look at meiosis. So, by the end of this topic, you should know what is meiosis, what are homologous pairs of chromosomes, and how does meiosis result in gametes. Just to recap, mitosis produces two daughter nuclei with the same number of chromosomes as the parent and as each other. So we say that they are identical daughter cells. So the number of chromosomes is 2n, n representing the number of chromosomes. They're exactly the same in the daughter cells as the parent cells. Now in meiosis, 4 daughter nuclei are produced, and these have got half the number of chromosomes as the parent. So we say that they are haploid gametes. So if you notice here, you've got 2n in the parent cell and n in the haploid gametes, half the number of chromosomes. Now in sexual reproduction, 2 gametes fuse during fertilization. In order to maintain the number of chromosomes in adults in a species, the number needs to be halved at some stage in the life cycle. This halving occurs as a result of meiosis. So here you can see that the number of chromosomes in the egg and the sperm are 3. What will the number of chromosomes be in the diploid zygote? It's going to be 6. So twice the number of chromosomes in the gametes. So what is a chromosome? Well, when 2 sister chromatids join together by a centromere, we'll call this a chromosome. So what is a homologous pair of chromosomes? All diploid cells of organisms have got 2 sets of chromosomes. One set provided by one parent, the other set provided by the other parent. This means that there are always 2 sets of genetic information for each characteristic of an individual. Any 2 chromosomes that determine the same characteristics are called a homologous pair. If you remember, humans have got 46 chromosomes. So how many chromosomes are in the gametes? There are 23. So we say that the gametes are haploid. They've only got 23 chromosomes. And the diploid number is 46. So meiosis must reduce the number of chromosomes by half. And then fertilization restores the number to the original number, 2n. If you didn't have meiosis occurring, you would have all the chromosomes coming from mum, all the chromosomes coming from dad, so you'd have too many chromosomes in the offspring. So meiosis ensures that the gametes have got half the number of chromosomes in each. When these fuse together, you get the offspring with the correct number of chromosomes. Now, before the process of cell division can occur, you need replication of chromosomes. So we say that this occurs in interphase. The new copy of a chromosome is formed by DNA synthesis during interphase, and the chromosome copies are called sister chromatids. Sister chromatids are held together by the centromere. So this occurs before mitosis, it occurs before meiosis. This is DNA replication. Okay, moving on to meiosis. Meiosis has got 2 nuclear divisions. There's meiosis 1 and meiosis 2, and these usually take place after each other. In meiosis 1, you have homologous pairs separating, and in meiosis 2, you have sister chromatids separating. Notice how the number of chromosomes is diploid in the beginning. After meiosis 1, it's still diploid, and it's only after meiosis 2 that it's haploid. Okay, let's look at meiosis 1. There are 4 different stages. You've got prophase, early and late, metaphase, anaphase, telophase. So how to remember is PMAT, PMAT. Prophase, metaphase, anaphase, telophase. Now prophase, metaphase, anaphase, telophase also occurs in meiosis 2. So we're going to start off with meiosis 1. Prophase 1. What occurs during prophase 1? The chromosomes shorten and fatten and come together in homologous pairs to form a bivalent. Crossing over occurs, and the centrioles, which are those little purple structures, migrate to opposite poles. The spindle apparatus forms. These are those represented by those thin purple lines, and the nucleolus and nuclear envelope disappear. This shows you how homologous pairs of chromosomes come together to form a bivalent. This is also known as a tetrad, And this whole process is called synapsis. So you have homologous chromosomes coming together to form a bivalent. Crossing over then occurs. So during crossing over, you have the homologous chromosomes forming a bivalent, and pieces of chromosomes or genes are exchanged. So notice in this picture you've got orange and red, and how they are swapping pieces of genetic information. So this produces something called a genetic recombinant in the offspring. So where the chromatids wrap around each other and attach is called chiasma. Plural is chiasmata. The chromosomes may break at these points and swap similar sections of chromatids with one another. This leads to genetic variation. So looking at crossing over leads to genetic information. You've got them wrapping around each other so that genetic information is swapped. So it multiplies the already huge number of different gamete types produced by independent assortment. Okay, metaphase 1. How I remember metaphase 1 is metaphase middle. During metaphase 1, the centromere is attached to the spindle, and the bivalents arrange themselves randomly on the equator of the cell, with each pair of homologous chromosomes facing opposite poles. During anaphase, the entire chromosomes separate while they're still attached by their centromeres. The spindle fibers pull the chromosomes apart. So here you can see a piece of sister chromatid exchanged during crossover in prophase 1. And notice how one of the homologous chromosomes is being pulled to a pole. In telophase 1, you've got microtubules pulling two sides of the cell surface membranes together so that the cell becomes narrower towards the center until the opposite parts of the membrane fuse to give two separate cells. In animal cells, we say that cytokinesis divides the cell into two. In plant cells, cytokinesis does not form or does not occur, so the plant cell goes directly into meiosis II. Okay, meiosis II. Meiosis II is basically a repeat of meiosis I, but you don't have any chiasmata being formed, and it's sister chromatids that move apart rather than whole chromosomes. You've got 4 stages, PMAT, prophase, metaphase, anaphase, telophase, and then at the end you've got 4 identical haploid cells. Starting off with prophase II. The nuclear envelope disappears, the nucleolus disperses, the centrioles replicate and move to opposite poles, and then you have the spindle reappears at right angles to meiosis I. During metaphase II, the chromosomes line up at the equator, their axes are right angles to the first division, the spindle is formed and attached to the centromere. During anaphase II, the centromeres divide and the chromatids are pulled to opposite poles by the spindle fibers. So notice here how it's the sister chromatids being pulled apart, not the whole chromosomes. During telophase II, the nuclear envelope reassembles, the chromosomes decondense, the spindle disappears, and then cytokinesis divides cells into 2. So what are the results of meiosis? You've got gametes being formed, 4 haploid cells with 1 copy of each chromosome, 1 allele of each gene, and then you've got different combinations of alleles for different genes along the chromosomes. So, looking at the difference between mitosis and meiosis, what do you think are the differences? Well, in mitosis, you have a single division of chromosomes and the nucleus. In meiosis, you've got a single division of chromosomes, but a double division of the nucleus. In mitosis, the number of chromosomes remains the same. In meiosis, the number of chromosomes is halved. Mitosis, homologous chromosomes don't associate. In meiosis, homologous chromosomes do associate to form bivalents in prophase I. In mitosis, chiasmata are never formed. In meiosis, you have chiasmata being formed. Mitosis, crossing over never occurs. Meiosis, you do have crossing overs. Mitosis, daughter cells are identical to the parent cells. In meiosis, the daughter cells are genetically different from the parental ones. And then in mitosis, you have 2 daughter cells being formed. In meiosis, 4 daughter cells being formed. So in summary, what have we looked at? We looked at the 2 different stages of meiosis, meiosis 1 and meiosis 2. And then the different stages within each, prophase, metaphase, anaphase, and telophase. You need to know these in detail. And that concludes our lesson, the end.